CGIL de Koning. Dr. de Koning has been a professor in the Department of Animal Breeding and Genetics at the Swedish University of Agriculture Sciences since 2010. He's had academic leadership roles, including head of section, deputy head of department, vice dean, and currently deputy dean. His main interest is to dissect the genetic basis of variation in complex traits and apply this information to breeding programs. His research covers traditional livestock species, as well as excursions to fish, mice, and plants, and the focus of his research includes health traits such as bone health in laying hens, lumpy skin disease in cattle, and African swine fever in pigs. So thank you for joining us, Dr. de Koning, and I'm going to give the floor over to you.
Thank you very much, and thank you all very much for having me. Now I should be able to change the slide. Yes, let's change. Okay, so I'm DJ de Koning, and the work I will present today is basically the result of a large number of projects and collaborations that I've sort of tried to capture into 3 parts. So genetics of bone health in laying hens, and basically our quest to find the best traits or the best time and place to measure it. And so, as I said, it's basically built on a number of projects with various funding sources. It's over the years that I've been in Sweden, we've had a continued collaboration here with the Rosen Institute at the University of Edinburgh, colleagues at Uppsala University, University of Granada, the company Lohmann Tierzucht, and a wide variety of funding bodies and groupings of researchers that have left their imprint on the project. So what we want to achieve really when we're working in bone health, which has been many years now already, is we want to get better bone health through selective breeding, but also more and more try to understand the biology behind bone weakness in laying hens. We know there is a genetic component, but we don't quite know that well. We can what it really is that makes it happen. And in this whole journey, we want to take the research from the lab closer and closer to the farm, and then back to the lab for further refinement. So this problem that we are addressing, avian osteoporosis or bone weakness, it's a welfare issue that has been known since the 80s, 90s of the previous century, so to say. When they were clearing barns of laying hens, they found that at least 90% of those hens during what they call euphemistically processing had at least one fracture. And they could also see that a proportion of those fractures had occurred before depopulation, so when the birds were still alive. If we look at different systems, because of course this is really not quite a welfare problem, and I often wonder or get the question, why is this not more in the top of our minds? And it is partly, I think, because these bone weakness problems are not restricted to a single housing system. They occur in all type of housing systems, including those that we as humans or as consumers perceive to be better for welfare. And there's been a more recent study last year in Denmark, got up to a figure as high as 70% of the birds having some form of bone damage at killing them at slaughter. So if we take a UK flock of 90 million hens or a Swedish flock of 7 million hens, that's quite a big problem. Just kind of a reminder, and also like the incredible level of scaling that happens in the poultry industry. When we talk about laying hens, we have they keep pure lines at the breeding company, and that's where they really do their selection. They have very specific housing systems and ways of testing these birds, but that's where the genetic improvement is made. Then we get the grandparent lines and the parent lines, and all this like couple of tens of birds on the top scales down to 8-9 million birds, and 30 million eggs at the bottom. So it's an incredible pyramid. And you can see that if something goes not so well in the top of the pyramid, that really has enormous repercussions. So it is really in this breeding structure we have to work. And I will also show that a lot of the research that has been done is here in this top part of the pyramid where the selection happens. A little bit about this bone cycling in laying hens, because when hens are in lay, they need to make eggshells, and of course they get calcium in some form through their food, and that gets into the bloodstream. Then some of it gets stored into the bone marrow, the hollow bones that birds have, because they have this is quite a unique feature for birds. They have hollow bones, the long bones, and they have this so-called medullary bone that they use as a sort of repository for calcium. So from there, when they need to make eggshells, they can recruit calcium and other elements from there into the bloodstream and then to the shell gland. And of course, when this process goes awry for one reason or another, that is when we can expect problems with bone strength. So when this work started with the colleagues at Roslin or at the Roslin Institute, they actually took birds from this top line here, from the top of the pyramid, so pure line breeding birds, to the Roslin Institute, and they simply started to select divergently for bone strength into a high line and to a low line, which was then still a very established way to simply start testing, is this a trait that is selectable? Is it a heritable trait, if you wish? And the answer was yes. This came out in 2000, where they really showed that, okay, so they developed a bone index where they, based on breaking strength, retrospective breaking strength. So you get eggs from all the birds, and then you kill them, you test the bone strength, and on the basis of that, you decide which eggs you're going to hatch for the next generation, because it's rather indirect selection, which is one reason why we're looking for better traits. So what they could see here, that okay, when they start selecting, indeed, there is a clear trend. And in that paper, the estimated heritability for this bone index is about 40%. So that's good news. It means we can select for bone strength. So what has that then? The logical thing, this was pre-SNPs and pre-G, was when this work was done, not when the paper was published. That's another story. So these lines were divergently selected first, and then they made an F2 cross between these and simply do a traditional QTL mapping experiment, where they identified a huge QTL on chromosome 1 affecting bone strength, both particularly in the tibia, but also in the humerus. So one is the leg bone, the other one is the wing bone. So that was interesting. And one of the first, this is sort of more of a close-up of that, where we have the long chromosome 1 here and statistical evidence for a QTL, and then we're sort of in this area. at that time I was still at Roslin, and we decided that one of the things we thought we really wanted to do was to try and fine map that QTL and find out what was happening there. So first we added a load of SNPs to that region and got a slightly sharper peak. This is still in the F2 cross. And then we went on to test on the pure line birds. Remember this, the cross was the basis from selection lines. The selection lines were derived from commercial breeding line. So then we went back to a 2006 population of the founder line that was sampled then. They had sampled 2,500 birds. We took the top and tail and selected 950 out of them for genotyping in the region. So now we moved from the F2 cross to a purebred population. And then if we have these SNPs in the QTL region, we could see that, yeah, there is clearly an effect there as well. Maybe not with the same huge significance that we find in the F2 cross, but there is a clear effect here. Then we, and if we look at that effect, we had sort of 3 SNPs that were in a haplotype together. And if we look at for a single SNP there that basically tagged all 3 of them, the difference between the homozygous for the A variant and the G homozygous was about 70 Newton. So there's 70 Newton difference in breaking strength with the heterozygote in the middle. So that was an appreciable effect. Then we wanted to find out a bit more, so we took some later generations. I think this is now the 2011, 2012, where we did genotyping on live birds so that we could selectively take live birds with opposing QTL genotypes and then do a gene expression study. So what you see here is a Manhattan plot, but it's not one for a GWAS, it's actually one for a gene expression study. And where we were very lucky here is that this is also the region of the QTL. And if we look at differential expression between chickens carrying the A homozygous genotype or the G homozygous genotype, we get an extremely significant effect here, and some others there as well that hiked along. So that was very, very exciting. That one is really like the, I think the expression value between them was at least 8 times the difference, if my memory serves me well. Yeah, 8-fold difference when we validated by quantitative PCR. And it found out that this gene has an impossible name, cystathionine beta synthase, or CBS, and it is very much a metabolic gene. It is not a development gene. It's very much something that's central in the metabolism. So that let us, okay, in this case, it is something in the turnover of homocysteine that is maybe not going so well. So we got that published some years ago. So that was nice that indeed, okay, we can use this approach to learn something about the biology. So then we wanted to look in some pure lines, continue doing the GWAS in the pure line, and that is published already in 2017. So I'm actually going into now GWAS in another pure line from the breeding company, which is the Rhode Island Red, and this is much more recent work by my PhD student Mohammed Salam, and he looked at the Rhode Island Red line from Lomanteerzucht and presented this at the World Congress, and now has a paper submitted on it. So it's a bit more recent, but it is the same population that was earlier published without the the genomics by Ian Dunn and colleagues, which sort of showed that basically the persisting of ley was not a major cause of bone loss genetically, but that there was other links that has been out for a while now. So in that study, where we are still again at the top of the pyramid here, but now in the Rhode Island red line, where we had 920 odd birds from Lohmann Tierzucht that have been housed at the breeding facilities, where they measured the birds and they did all the standard measurements on the chickens, the bone density, the breaking strength, the medullary score, and body weight, etc. Then the bone samples were sent to University of Granada, where they did a lot of work on chemical composition on both the cortex and the medulla, so the two parts of the bone, the medullary bone and the cortex around the outside, where they measured bone composition, both using infrared approaches and thermographic measurements. I will get back to that one. And then we genotyped them for GWAS as we or with our colleagues at SciLife Lab in Uppsala. So this thermographimetric analysis, TGA, it's an interesting measure. It's kind of like, not familiar, it's like a heating ramp, and it measures different bone components by weighing them as the temperature go up. So first you lose the water, and bit by bit different components of the bone disappear, and you sort of quantify that by weight. And this was done separately, as it says here, for the medullary and the cortical bone. And then we aim to correlate that with other measures and mechanical load of the bone. So the breaking strength, see if any of that is a good predictor. GWAS analysis were done by Mohammed Salam in JAMA, mixed linear model, where the hatch and the house of the birds were fixed effects, body weight as a covariate. As we'll see later, body weight has a very strong link with breaking strength. So if we're not careful, we're just mapping body weight QTLs. SNPs as a fixed effect, and hens as a random effect with a genomic relationship matrix. And then also did some other modeling for correlations using the marker data using GREML. So straight into the GWAS results. So we see for cortical lipid, this is the most significant result. We had the conservative Bonferroni thresholds, blue, suggestive, red, that we call significant. And chromosome 3, there seems to be quite a strong effect on cortical lipids, so the lipid content in the outside of the bone, the hardest part. Something even for the ratio between two bond calcite and amide 1, another ratio between phosphate and amide 1 in the medullary bone, and one for the collagen maturity. So some not super convincing, but at least some hits coming up there for bone content traits. And there's more here, even an effect on chromosome 1 again, another one on chromosome 2 for the crystal scattering and the orientation. So these are more microscopy traits. And finally, breaking strength, unfortunately, not showing a really convincing peak here. So there's not a major QTL for breaking strength itself in this population, but we do find some other hits. And if we look closer at the best one for cortical lipid here on chromosome 3, when we zoom in, where it's just the QTL region, and then the dots are colored according to the LD with the best SNP, and the same here on chromosome 2. And basically what this points to is, okay, there is a cluster of significant SNPs, but they're all in extremely high LD. So this at least suggests that we are looking here only at a single QTL, both on chromosome 3 and chromosome 2. And Salam has looked a bit further into potential candidates in this region, and sort of that's something that will come out in the paper. But again, there's some potentially interesting candidates, but nothing that jumps off the page. What we also wanted to know, though, okay, now we're looking into more detail. We want to learn some biology here. Is there anything here that helps us explain breaking strengths? Which of these correlates, and let us learn something about breaking strengths? And then even there, we had remember we had the most significant QTL for lipid in the cortex of the bone, and even there we find the highest genetic correlation, which is not massive, it's 0.20, but it's a negative correlation, so higher lipid content gives lower breaking strength. So that was the most notable one from those correlations. And then we also wanted to compare to studies between other components from the medullary bone that showed some significant results, the mineralization of the medullary bone and the overall breaking strengths. These again were very modest or even non-significant. Egg mass, Ian Dunn already showed that had a significant correlation with these traits, and we find basically non-existent correlations with this cortical lipid trait. So we're trying to find sort of how does it all hang together, but the answers are not really staring us in the face, unfortunately. So how about, now we have looked in crosses, we've looked in pure lines, how about commercial laying hens? And there we had a big study that we did ourselves in Sweden with a lot of collaborators, and we were very happy to publish that recently. And that work was pulled together by Martin Johnson, who is a researcher in our department, and very good researcher. I can very warmly recommend him, but just don't recruit him away from us, please. So that we want to look at commercial laying hens, because it's all very well to find QTLs in pure line. In the end, they need to have an effect in the commercial lines to make any benefit for breeding and for production. This work was published very recently. So yeah, you can find QTL, but what's next? Well, what we really wanted to know, and we had some other studies published earlier where we do find significant QTLs in these pure lines, but what is their meaning in commercial layers? Do they have any sort of role there? Is there an effect of the housing regime on these QTL or on the breaking strength? Is there an effect on diet supplementation? But also, is there an effect on the appearance of the bird, the feather score, for instance? And the part on the feather score has been published by Helena Wall, and I will not discuss that here. That paper is out there. And just a reminder, so then we move in the breeding pyramid to the bottom here, where we are simply using commercial layers. So what did we do? Well, we used commercial layers from two lines that are commonly used in Sweden, from Bovans from Hendricks and Lohmann selected Leghorn from Lohmann. These are white chickens that are preferred in Sweden mainly. And then we needed to find a hatchery that could hatch different breeds and raise them in different systems. That all worked out. And then we had in our own experimental farm near Uppsala, We had the birds in 2 housing systems, enriched cages with 8 birds per cage, and floor pens with about 100 birds per pen. 2 levels of a zinc feed supplement, and we filled that barn to capacity, which is not that huge. It's 3,500 birds, but simply to mimic real life as much as possible. And also we extended the experiment to keep the birds to 100 weeks of lay. So if you want to about this integument scoring, we really the birds were regularly scored for their appearance, and this is according to the welfare quality 14-point scale that you can read about here. And we had one experienced technician who throughout the experiment looked at all the birds using this approach. But at the end of the experiment, we couldn't use all 3,500 because of logistics. It's a lot of work to get all these birds dissected and sent away for analysis. So 800 birds were scored for feather score and keel bone. Then they were put down. And then if they were still in lay, then one tibia was kept in Uppsala for bone strength and radiography at the University of Uppsala. One bone went to Granada to do bone chemistry there. Colleagues from Linköping University had an interest in comb, so they took the combs from a number of birds, and we took from every 5th bird a bone sample in RNA later and one in -80 for potential gene expression studies. And in this case, we had to extract DNA from the liver. So in this bone scan measurements, the CT measurements that they did in Uppsala, we get this PQCT, and it's kind of helpful to illustrate that if we have the bone here, then one cut is made here halfway, and one cut is made there. And where it's then being very scanned in lots of detail, where we get the medullary bone and the cortical bone and all these different measurements that can be determined in such a 1 millimeter slice of bone. And we were hoping also here to not only learn what bones were simple breaking strengths, but also how would these different bone component traits, biomechanic traits, correlate with breaking strengths, hopefully learn something what makes a better bone. More about that later. So we did analysis in GenStat or in Emacs when it comes to GWAS. CT measures were highly correlated, these PQCT measures, so we decided to summarize them as principal components. We did the association analysis within the breed or the lines, the bovine separately from the LSL, and used general linear models with effects of breed, housing system, feed supplement, etc., and weight as a covariate. So in terms of the experiment, we had for body weight 440 birds from the pen system and 492 from the cage, so we got more than we planned for, and for body weights very much comparable numbers. So first of all, we start to look at the breaking strength in these different systems. If we look here in these scatter plots, we have here the cage birds and the birds in the pens, and the same here. Here we look at body weight, and all the red dots are LSL and the blue dots are bovines, and we see there's not much difference for body weight between cage and pen, where the maybe the one breed is slightly bigger, but not all that much. But we see for breaking strengths, we already see that there's a big difference that the birds in the pen have stronger bones than the birds in cages. That's quite easily explained because they move a lot more, but we also see a much bigger spread. Not a big difference between the breeds here. And that trend really continues. I mean, we see here, we plot body weight against tibial breaking strength in cages or pens, and this is the regression lines. We see that the regression lines are almost identical within the breeds, but we see that the line is quite a bit steeper in the pen compared to the cages. This resulted in the fact that we needed to basically, for further analysis, treat even cage and pen. We had hoped to just be able to stick it into the model as a fixed effect, but now it basically meant like, okay, we're going to need to treat this as two different traits. Because this housing system and breed keep cropping up. When we look at these bone content traits again, proportion phosphate of total organic material in the medullary bone, very big difference between cage and pen. Again, for the medullary phosphate percentage and the medullary range between ratio calcite and phosphate. Sometimes even a difference between the breeds, but always the difference between housing system being a pain in the body part. Okay, so all these bone phenotypes that we measure, this is a very busy figure, but here we have all the CT traits, all the PQCT scanning traits that are here, and they cluster very tightly with each other. They cluster actually extremely tightly with each other. Here we have the medullary bone composition traits and the cortical bone composition traits, and also they correlate quite strong with each other. And what we see here is our favorite trait, of course, bone breaking strength, and here also bone breaking strength. Clear correlation with body weight there, but basically bone breaking strength here, or whichever way you look here, correlates very strongly with all these CT traits, basically to a degree that we don't learn much new when we look at these bone composition traits here. It's a little bit better when we look at the chemical composition traits, where at least the cortical composition shows some relevant correlations with the breaking strength, but not so much so the medullary, it does not have a very strong either to the QCT measurements or the breaking strength. So yeah, we did not learn as much biology here maybe as we hoped, but at least we have traits, and give me traits, give me markers, we will do GWAS. But as I said, we already knew that we had to do it within breeds, then we also had to do it within housing systems. So then our power isn't so fantastic anymore, but at least we did get some suggestive results for bovines in cages, maybe even some from LSL in cages. And when we put them together, we don't tend to get much when we pull across housing systems. But for comparison, if we look at another trait like body weight, then actually we get quite strong links. Here we have an effect in the cage in the bovines, maybe not as strong here, but if we put them together, is a very strong peak. And likewise, if we look at the LSL for the chromosome, well, it's the chromosome 4 QTL in chickens that props up here. So we have an effect here, we have an effect here, and if we put them together, so the body weight QTL seems to be quite consistent across housing systems, but the bone QTLs are not. Then we have some of these more esoteric traits like the quantitative, the PQCT, the scanning traits, and one of the principal components that could be characterized as low cortical density, sort of the common denominator that showed some effects here in cages, on bovines here, and in LSL also on chromosome 4, could well be the body weight QTL. But again, no major findings. And it's kind of, okay, we don't have the best power, but we're kind of triggered by this. Is it really a G by E? And I think it's quite clear when we look either at the P values or the marker effect. So on the top, we see the bone strength, the P values, either the P values for a given marker in the pen system, plotted against its comparable log10 P value in the cage system. And it seems almost inverse, like the ones that are relatively high here are very low in the cage system, and the ones that are sort of interesting in the cage system don't show very much in the pen at all. So it seems almost non-opposite. And the same here when we look at the estimated marker effect, an estimated marker effect in the pen here, and the corresponding marker effect in the cages there. So it's not much, and I'm admitting we don't have fantastic power, but if we compare that for the corresponding values for the body weight QTL, then we start to pick up some here that at least are clearly significant in both systems. And also, I hope you agree with me that we have a little bit more consensus here when it comes to the actual marker effects in the both systems compared to when we contrast the effects of the different systems for bone strength. So what have we learned here? Well, basically that whether we measure it from birds in cages or birds on floor pans, we get very different results. There's a very big effect. And that probably means that selecting in the pure lines, like in the breeding company where the birds are kept in cages, will not maybe get me the best bird's bone strength when they are living in pens or aviaries or free-range systems. So we probably need to be measuring more at the farm level. So that takes me to our current project, the Keel Bone Tools project, funded by the Swedish Research Council Formas, where we really want to go to more the target. A lot of the welfare issues and a lot of the research funding, admittedly, is aimed at keel bone damage in laying hen. That's a major part of bone health. So why do we study tibia all these years? Well, basically because it is a lot more straightforward to phenotype. We have clearly established protocols, and we can repeat it study after study and get a pretty good, reliable phenotype. Also, what we found on the tibia, we get really good heritabilities, 30-40%. So we like that. We didn't hit gold yet with anything on the keel bone itself. So we always expected, well, good bone health overall. If it's a physiological property, then good bone health in the tibia will translate to good bone health in the keel bone. But really, if we want to improve the keel bone, we need a good high throughput phenotype. And one of the suggestions that's been made by Christina Ruffner and Mike Toscano and these people is really we should be able to, from palpation, why don't we get a live hen phenotype by taking X-rays. And there was a lot of that done in the killbonedamage.eu cost action. And Ian Dunn has done sort of implemented that in the pure lines, so in these selection lines at Roslin, the breeding lines from the breeding company in a US-funded FFAR project. And they recently published this sort of X-ray method and how to do it, basically. That paper is out recently and got some attention. So again, what we want to do then is take it from the pure lines to the commercial layers, but not just take it to the commercial layers, actually take a step further and take it to the farm. So now we would like to introduce X-ray as a phenotyping tool at the farm level, basically test whether it's sort of, what does it involve? It's a tool maybe for monitoring. It could also be a tool to provide crossbred information for purebred selection. And as we're going to take a huge load of X-rays, can we, maybe this is sort of a hope, further away hope for the project, can we actually use some of these artificial intelligence tools to do automated scoring of X-rays? So we really want to learn from this cost action where we had and also see if we can find a working phenotype for the farm. But at the same time, of course, take all these birds that we do and dig into all these other measures that we have looked at over time, trying to understand bit by bit what is happening. So first of all, we take this one part of this project to simply take the FFR project that Ian Dunn led from Roslin, add genotyping as an extra layer there because that was not included in the original project, perform an additional analysis on these bones. So for this project, the bones have also been shipped to Granada for analysis, and then see if we can do this GWAS on keel bone damage as assessed by X-rays. Combine that with other bone traits and really see if there is ways that we can get good predictive value from these X-rays. What we're doing in our on-farm study that has happened in Sweden is, as I said, a regular Swedish poultry farm not far from Skara. In that poultry farm, we sort of fenced off a bit inside the barn, where we follow a cohort of about 200 hens with regular X-rays over productive life. And we need to really, this is a disturbance, so we really need to minimize that and think, okay, how often do we go in and how do we work that? So then we hopefully have good longitudinal data on a number of these animals. We also, from that farm, from each batch, we collect usually 100, but finally 500 batch, just basically kill them and store them for post-mortem X-rays, followed by dissection. So these birds are now in the freezer. We will subsequently X-ray them, then dissect them, measure all the bone characteristics and see what can we learn from the X-ray or that correlates well with what we find on the dissections. Of course, full genotyping. Again, the bones will be sent for detailed phenotyping in space. And this experiment was finished just before Christmas, when the dissections are now imminent. So that's work in progress. What have we learned so far? Well, we find a pretty strong suggestion for gene by environment interaction for housing systems. And that has consequences for how do we do our breeding for better bone health in laying hens. Apart from some excited work that we did earlier, no really major genes or silver bullets. We're still searching for a keel bone phenotype that really scales for genetic evaluation. That is really key. If we want to improve it, it needs to be something we can do routinely. And I think we're really going to need farm data from crossbred layers from different housing systems to then link that back to sort of as a training data for selection in the purebred lines. And that is something that Salam has recently worked on. Is basically from the data we have, how well does it cross-validate? How well what do we need to get a good genomic breeding value for these crossbred birds? This is the team that's working very hard in this project at the moment. It's myself, Martin, Helena Wall from our sister department, Stefan Gunnarsson, people that help us on the farm, Anna Larsen, Lina Göransson, my student Salam, who's fantastic, Ian, done at Roslin, a long-term collaborator, and what Ian doesn't know about bird physiology is simply not worth knowing. And likewise, Alejandro in Granada, he has phenomenal knowledge about bone biology. So with that, I thank you very much for your attention, and I'm happy to entertain some questions. But I will stop sharing so I can see you all a bit better.
Thank you, Dr. de Koning. If anyone has questions, you can raise your hands, turn on your microphones and cameras if you please, or you can enter your question into the chat as well. I think there's one in the chat at the moment, if you can see it, or if you want me to read it to you.
I can open the chat. Do you have any data on Japanese quail? No, I do not, but I'm sure there may be some. I had a really, related to that, had a really interesting conversation with a PhD student from Germany earlier last year who works with Professor Dirk Indriks. She had been part of dissections of traditional breeds because we think this is a problem that has occurred in poultry breeding, right? And they actually found that in traditional breeds, the keelbone problems are also highly prevalent. So it's not even something that is that new. So that's kind of, in a way, reassuring. In a way, it's okay. It's a widespread problem, but it's not simply breeding has brought this about.
Yeah, I was wondering, oh, okay, let's go with Colin.
You can go first if you want, Kristen. I don't mind waiting.
I was just going to ask, because you mentioned that that breeding may not have caused this, but that is kind of a common thing you hear, is that breeding is the cause of these problems that are occurring in these animals. So do you know kind of maybe what has caused it, or if it's always been a problem, and if we know, yeah, I guess just the cause of
so
this.
we what has really caused it, we don't quite know, but what is emerging, especially from the paper that Ian had in 2021, that is cited in the presentation somewhere, that it is one part when birds do better is if they enter into lay a little bit later. And that is not so strange, because the keel bone is one of the last bones to ossify. So actually, if the age at first egg, if that is a little bit later, then they have they're better equipped for their productive life. Another part that's very important is to raise the birds in the system that they will be producing. That's also quite a clear advice. It's not actually the length. It doesn't tend to be the length of the lay that in the end they will all get weak bones. It's basically so things that happen early in life tend to be more important than things that happen later in life. And of course, what we see is that birds that are in these more open systems, they get better bone strength. Overall, their bone health is better, but they also create much more opportunities to hurt themselves. Colin.
Yes.
That partially answered a bit of my question, but my kind of question centers around, you had mentioned that there was no effect of laying persistency on bone quality. Was I correct in my hearing with that?
Yes, that is from the previous studies, that they once you get down to the genetic correlations, there is no genetic correlation between persistence of egg laying and getting weaker bones. There may be a phenotypic link, but that's not. It's not that it's linked in the sense that, oh, if you select for one, you will automatically get the other one.
So my question kind of around that then was egg size looked at all? Like, was animals that had a predisposition to both more, like what I'm thinking is like calcium into their bones may have smaller eggs compared to other ones where the persistency wouldn't in itself be affected, but the actual size of the eggs may be affected.
So egg size has an effect, but I'm not sure that's part of that. Could also be when we think about the keel bone and where the egg sits in the chicken, that it actually could squeeze more towards the keel bone, so it could actually be one stressor. And so rather that a big egg could be an internal stressor on the keel bone, rather than a physiological correlation between calcium in the egg and the strength of the bone.
Okay, perfect. Thank you.
Emily, do you have a question?
Hi. I was just wondering if in your studies where you were looking at cages versus pens, if they had the same types of furnishings within them. So like if you had perches in both of them, or were there no furnishings?
Yeah, the cages that we used were the so-called enriched cages that have a little scratching area. They have a little perch, so that's. But of course, the floor systems, by their very nature, had a bit more entertainment because they had laying areas and sort of, yeah. So the paper by Helena Wall that I haven't put in the presentation, but her work describes the housing system very very detailed. If you're interested in that.
Yes, I will have to look that up. That's very interesting. My research kind of looks at that as well. So
cool.
And there's a couple.
Look at look at the questions in the chat, maybe.
In the chat. Yeah. There's one from Rick. He kind of asked the same thing as Emily. And Christine has a question as well.
All right. How expensive is the phenotyping on a per bird basis, and are there any other potential indicator traits? How much is the phenotype? That's a good question. It's so it depends how you count it. I think the TGA is fairly. I think that's a lot of bang for the buck in terms of equipment, and it's mainly staff time. So I think that's a pretty good one. I think the CT scanning. I mean, I don't have exact figures on these, but the CT scanning is of course more expensive. It's quite a specialized equipment and quite involved. Other potential indicator traits, that is a little bit where we're looking with this, hoping to get at with these X-ray things. Of course, for keel bone, you have this palpation scales, and you can get trained palpation scores that only cost the time of the person that does it. They are pretty good indicators for a farm how things are going, but they don't tend to do very well on sort of when you want to take it to estimating heritabilities or something like that. So at the moment we're still looking, and I know they have Heather and from Roslyn, Heather and Bob and colleagues at Lohmann, they have really been looking at, okay, can we do a quick ultrasound? Ultrasound has been tested quite a lot because that would be a lot quicker and a lot easier and not very invasive at all. And it hasn't really worked out. So they've looked at ultrasound a bit.
Thanks so much, DJ. That was really great.
Okay, I think that puts us pretty close to time. If there's no more questions, oh, Flavia?
Yes, I have a question, Dirk. Thanks
Hi,
for
Flavio.
the presentation. I don't work with birds, but what I was thinking is that if the Q bone, if I understand in the bird, the Q bone is where the major muscles from the wings attach, right? So it's very important for the flying of the bird. If the domestication of the birds, we don't allow them to fly anymore. So they are not flying birds anymore. We will not expect that this bone will get weaker because it's less functional now in terms of the important muscles that attach to it. So,
yeah, that's.
So I think more than selection, I think the domestication of the birds in general would cause part of this problem. Of course, could be more intensified now because the animals grow faster and so on. But the fact that don't fly, I think, is something that.
But actually, in the very good point, Flavio. But in the more open systems, the tiered systems, and where they have perches, they do actually fly a bit. And there's actually been work from colleagues in, I think, UC California somewhere, where they actually, and there's actually one working hypothesis whether some of these damages they see on the keel bone is not a collision break, but actually a stress fracture when they try to navigate steep inclines, or like they have or person where they sort of, so they do use their wings for balancing if they're not flying, but some of them do sort of fly a bit. So they're actually looking into whether part of it could be stress fractures rather than collision fractures.
Yes, yeah. No, that's what I think. They stress because they are not using fully anymore these muscles, right? And therefore I don't know, they evolved to a weaker bone. But anyway, I was just curious.
Interesting, yeah. It's an interesting note.
Okay.
Good.
Yeah, thanks for the presentation.
You're very welcome.
Really good. Thanks a lot.
Okay, I think that puts us at time. So thank you everyone that joined us today. And thank you, Dr. de Koning, for the amazing presentation.
Thank you very much for having me, and have a great remaining Friday afternoon.
Yes, you as well. Have a nice weekend.
Hi, Christine. Good to see you.
Hi, DJ. Thank you so much. Super interesting.
Good. It was a joy. Thank you very much.
Thank you.